Hello, my name is Jorge and I'm a Product Support Manager at Biognosis. This tutorial will guide you through all the spectrum of features that support DIA analysis of isotopically labeled samples. As exemplary data, I will be using a Pulsar dataset which was published alongside an article by Barbara Salowska from Janssen's Lius lab. This publication is an interesting read and we will highly recommend it if you want to learn more about the settings and plots covered in this tutorial. You can access the publication directly through the link in the description. Let's walk through the steps to properly set up a direct DIA analysis using a labeled workflow together. While I will focus on settings relevant for Pulsar data specifically, I will make sure to point out relevant changes for other labeled workflows, including the use of heavy spiking peptides. This dataset is comprised of two different fibroblast cell lines labeled with heavy lysine and arginine over a 24-hour period, with samples collected separately at 4 different time points. Starting from the default settings, first you should define your channels in the labeling node. While this experiment only has 2 channels, Spectronaut supports the use of up to 5 different channels at the same time. The setup of this experiment requires that we use a light channel where no labels will be specified, and a second channel where we will add our relevant heavy labels. Keep in mind that, when utilizing the labeling workflow, Spectronaut assumes that all specified amino acid residues for a given channel will be uniformly labeled. In our example, if we had 2 lysines within a peptide sequence, both will have to be labeled to identify this precursor in channel 2. Moving now to the workflow node, it is recommended to enable the setting in silico generate missing channels to ensure a homogeneous set of channels across all samples in the generated search space. When selecting a labeled workflow, Spectronaut will in silico create any missing light or heavy counterpart for all identified precursors. For spiking experiments, this workflow will generate a light channel for any heavy precursor for which a light counterpart was not identified. The inverted spiking workflow generates missing heavy counterparts for all identified light precursors. In the Result Filters node, selecting the setting option Overlapping between channels will exclude fragments shared between channels from the search space. This means that such fragments will not be used for both identification and quantification purposes. This is important for channel-specific FDR filtering. If this setting is left unchecked, shared fragments will still not be used for quantification, but could be used for peptide identification. Let's move now to the quantification node to discuss the multi-channel Q-value filter. This setting allows users to specify how multi-channel precursors should be treated for channel-specific FDR filtering. To demonstrate the impact of each option for this setting, let's look at Figure 1 from Salowska's paper. When the group Q-value option is selected, a given precursor pair, labeled and unlabeled, will be considered identified and subsequently quantified only when the Q-value calculated by combining observations from both channels overcome a defined threshold. Alternatively, selecting the mean Q-value option means the pair will be quantified when the precursor in just one of the channels is independently identified. Lastly, the max Q-value option means the precursors in both channels must be identified independently to be quantified. Lastly, in the Workflow tab of the DIA Analysis section, the multi-channel workflow definition should be adjusted to match your desired workflow. The From Library Annotation option will match the workflow definition from the Pulsar search settings with your selected fallback option. In this example, the fallback option can be left as labeled. When setting up a spiking experiment, you should select Spiking here, which enables several additional options. The target is the channel we would like to identify and quantify using spiking peptides. Therefore, it is the target channel that determines the identification status of a given peptide. The reference channel is a term used for the spiking peptides and is used for peak detection purposes. By default, the light channel is considered the target and the heavy is the reference, but users can invert this by clicking the relevant checkbox. Moreover, for both spiking and inverted spiking workflows, you can choose the option to perform identification on the reference channel by enabling reference-based identification. Regarding condition setup, users can assign various conditions to different channels for the purpose of differential abundance analysis. The condition setup allows also convenient sample-specific channel condition assignment, which supports labeled workflows with swapping the labels. Examples of common condition setup configurations for multi-channel experiments can be found in the Spectronaut user manual. Now, let's view the results of this analysis and discuss the visualization and reporting options. In the analysis perspective, when selecting a protein group, you can visualize the heavy light cross-run ratios plot. In this graph, intensities of heavy and light precursors are used to calculate a linear fit where the slope represents the protein heavy to light ratio. Conditions and colors can be modified in the condition setup interface. In the post-analysis perspective, you can find two additional plots that are relevant for this kind of experiments. The scoring weight per channel plot shows an overview of the contribution of the different channels to the overall scoring in different runs. The Pulsar heavy light ratio is a convenient way to check the ratio of heavy versus light intensities across different samples. Additionally, for experiments designed for this purpose, you can perform the differential abundance analysis across channels. In channel-based differential abundance analysis, results will be reported in the candidate table and visualized in the volcano plot. Lastly, let's go through the reporting fields available for multi-channel experiments. Information on quantities at the precursor, peptide, and protein group levels are available for the different channels. For Pulsar, experiment quantities at the precursor level can be reported using the field FG quantity. The field FG label sequence will give you information on which channel a given fragment group belongs to. A tip for navigating the different available fields to be included in the report is using the search bar located at the bottom of the window. If we type channel, we can see that quantities for peptides and protein groups based on MS1 and MS2 signal can be reported independently for each of the channels, as well as channel-specific Q values. For spiking experiments, you can report precursor quantities for your target and reference channels using the fields EG target quantity and EG reference quantity. Additionally, ratios between the channels can also be reported using the fields EG heavy to light ratio when using two channels, as well as target to reference if you are using two or more channels. I hope the information presented in this tutorial helps you set up the analysis of your labeled samples. If you have any questions, please reach out to our support team. Goodbye for now.